Hello. And here we are. You ready, Chris? Welcome to Connected Conversations, and we're going to have a great chat tonight with Chris King. And you're one of the people I just kind of found on Twitter one day, just kind of snooping around, and as I tend to do, and a lot of people on Twitter, and I came across your artwork, and I was really curious about how these images came to be. So I wanted to invite you on the show to talk about that. I know there's a lot of interest in the intersection of art and science. Ben Games, thanks for stopping in, buddy. Good to have you here. I haven't seen you in so long. I got you here with the art. So Chris, if you could just talk about who you are and your background a little bit to introduce us to you.
Sure. Thanks for having me. I'm Chris King. Basically, my hobby is using my microscope to make kind of psychedelic, interesting crystal artwork using various chemicals I find online or stuff I use around the house. I mean, I've always been interested in science. I kind of didn't really know what I wanted to do back in high school and college. I really liked biology and stuff. And when I graduated college, I just got kind of a temp job, I guess, working in various labs. And I ended up working in a pathology lab. Did a few different sections in there, and eventually ended up in histology, which is the study of tissues and how cells are organized together, which is mostly done through the microscope. And you know, some days I'd have a lot of downtime at work, and I was researching a new staining procedure we learned where we use different chemicals to color different parts of the tissues to make them easier to diagnose diseases or different defects, cancers, infections, that sort of thing. And one of our stains is called Congo Red, and it uses what used to be like a textile dye people used for clothing until they discovered that it was very harmful to have the clothing on your skin because the dye actually reacted very strongly with people's tissues. But it makes it a good pathology dye to look at, I guess, scar tissue and tissue damage. But the trick with that stain is that you can cut the tissue pretty thick, about 8 microns thick, a little thicker than like a half of a cell, I guess. And then you use polarized light, and it makes different parts of the tissues glow. And the dye sticks to the scar tissue and makes it glow green. I thought that was kind of neat because I'd never really heard of I've heard of like polarized sunglasses and that kind of thing, but I didn't know much about polarized light. So I started looking into it more online, and I found that there was a whole various uses of polarized light besides just to visualize scar tissue for this one stain we were doing. People used it to look at thin sections of fossils and minerals and geology, different kinds of plastics to look for like structural defects in the manufacture of them. And some people just like making very interesting crystal landscapes under the microscope, real thin, but the way the polarized light kind of illuminates the crystals gives a really interesting depth and color to it. And I found we had a bunch of chemicals in the lab we weren't really using, so I started playing around a few days of work.
Some people call it playing around. Other people call it science.
Yeah.
I mean, essentially that's what you were doing. And this is, I have some of my students are actually in the chat. At least one I know is one of my students. And, you know, one of the things I try to tell students, and especially 13-year-olds, it's kind of hard for them to see in the future that you never know what you're going to use will apply to something else, right? Like the problem solving that you kind of used in determining your art, you know, at some point, you know, you had the logic and the reason to be able to like do trial and error in a good way to get to a solution for what you wanted to come up with as far as art. And I don't think people usually make that connection between that reasoning skill and art in trying to do that trial and error. So, and you never know, like you started off, not thinking that you're necessarily going to go into doing these techniques, but here you are.
Yeah. I thought I was going to do kind of like a David Attenborough, Marty Stauffer thing and do like wildlife documentaries or something, and then totally different field, but the appeal of the kind of artistic side of things, because I already liked making nice looking slides to look at under the microscope with the tissues we prepare to work. So
yeah, I mean that's part of the job too, right? I mean, I don't know how it is. Would you get rated, or your evaluation would be on the quality of slides that you could produce for the histology? Is that?
Yeah, the doctors let you know if they're not happy with the quality.
Oh, I'm sure.
Very, very picky.
Yeah, I mean that lends to, refining your technique, for sure.
Yeah.
So how long have you
pictures?
Yeah, how long have you been doing this now?
I think it started back in 2016. I mean, I was already kind of taking pictures of some of the slides I did at work, because it was really hard to explain to my friends and family what I actually did during the day. Mm-hmm. So I'd kind of put my cell phone up to the microscope lens and take pictures of like bacteria and stuff we stained under the microscope. And then once I started playing around, or experimenting, I guess, with some of these different chemicals and making thin layers of crystals, started sharing some of those too, and people got pretty excited about it. So I was like, I guess I could make prints of some of the good ones. And
and you have, I know you have some up on your link on your Twitter and on your Instagram as well, for prints. So people can definitely contact you if they see something that they like. And I read through a lot of your bio information, and you have a lot of skills in biology. And like I'm looking through the different things, and like a lot of it involves dead animals. Don't get me wrong, that was kind of like, okay, but someone's got to do that.
Animals.
I was the guy in lab where no one wanted to euthanize their mice so they could dissect their brains or whatever, and I was that guy. It's like, Scott, can you kill my mouse for me? Can you pith my frog for me? That was me. Someone had to do it. I got those skills.
I was always that guy for like the pig dissections and stuff.
Yep, you were the same person. So I kind of thought of that when I was reading that. I'm like, we got something in common. We're the person that does that. Not the pleasant thing, but science.
No, I never really enjoyed the euthanizing part, but you got to do it sometimes.
But I mean, you are an animal person though, so because I notice you have a lot of experience and not necessarily like zoologist kind of stuff, and not necessarily veterinarian, but you do have experience with animals. Do you have a lot of pets? I saw a cat back there, I think, once.
Yeah, I got 3 cats now, and I'm watching another one. There's going to be a 5th one coming on Friday, so it'll be a pretty wild house. I had a dog too, but yeah, she kind of passed away in December. She was old. I've always loved having pets and stuff. I grew up in the mountains in Pennsylvania, and we always had animals around, and wildlife, bugs and flowers and things I like to look at. And my aunt had a sheep farm, so I was always going over there to help out with the sheep and her herd of cats and horses and stuff. So
I did spend a summer in Warren, Pennsylvania. That was my one time I lived in Pennsylvania. Down the Allegheny.
Sadly, I haven't seen much of the state actually, but I think I know where Warren is.
Top left.
Gotcha. Yeah.
In the middle of nowhere. There's nothing there.
Yeah, Pennsylvania's surprisingly rural. There's only a few cities there.
Surprisingly rural and really bad roads, usually.
Yep. Yeah. Terrible roads in Pennsylvania. Once I crossed the state line into Maryland, I'm like, wow, I see why we pay so much taxes here.
So let's get into some of your artwork. The
Sure.
one that we have up right now, I was saying earlier how I feel like I'm kind of getting sucked into it. It's like that part of a movie where like the character is getting drawn through an interdimensional pathway or something.
Exactly what I was going for.
How well, that's what you got. And I know art is good when it elicits an emotion or a connection like that, and that's what I liked about this one and a lot of your other ones. There's kind of that, immediate connection to it. Can you describe the process you went through for this one in particular?
Yeah, this is one of my favorites. I actually did it a few years ago, but it's the chemical is dopamine hydrochloride, which is it's sort of like the dopamine you have in your body to, help your muscles move and elicit the feelings of reward for doing something cool, like listening to music or having a good time. But the hydrochloride ones, they injected to people that have like low blood pressure, like for shock and stuff. So anyway, I pretended I was a research organization and got a bottle of it online, and so I took like a tiny, maybe 5 milligrams of it in a little scoop and put it on a slide and heated it up over a hot plate with a cover slip on top. And the center of the image you can see, but kind of sucks you in. I had a forcep kind of holding that part down, and so that sort of caused like a nucleation point. So the crystal started growing there and then spread out. So the thinnest part of the crystal there is that central point, and then the colors kind of change as it gets thicker coming out. The polarized light can kind of tell you how thick a sample is based on the color you get from it.
Okay, so the color is based on the thickness, and that's within the polarized light, and that's how the effect is. And then is it just by chance that you get this? I mean, how much of this? Because it's cool in that it's like you have the orange in the bottom right corner, you have the green in the upper, you have that orange yellow again in the top left, and then the purple in the other corner. I mean, that to me that seems like it's something hard to get it to crystallize the way you want. I mean, is that the case? Or can you guide it by like scratching the surface or something like that?
Yeah, you can kind of manipulate it through either applying pressure, or if you're like evaporating a substance, it'll kind of enter like a gooey phase or liquid crystally phase, and you can drag something through it, and that'll usually get the crystals to start forming where you disturb it. Well, the other thing you can do, like on a polarizing microscope, the stage, instead of being like a square when you move back and forth, it actually rotates, and as you rotate it, different sections of the picture, the colors will change, and sometimes darken or lighten. And then I got a couple of extra filters on my microscope that I can slot into place to kind of adjust the colors. Or you can put we call them light retarders, which is kind of an unfortunate name, but they slow down the light going through that. So if you put one of those in the light path, you can kind of get more fine control over the colorization. I assume they have some analytical purpose, but I like to use them to make the pictures look kind of prettier, more or less colorful. But yeah, it's a lot of, I'll make like 30 slides of the same thing until I get like a couple of good pictures.
Now, how much of this is still kind of experimentation with you, and how much of it is you kind of at this point, after doing this for 4 years, know how to get the image that you want?
Kind of depends on the chemical. Like some I've done a lot more trial and error until I kind of know exactly what the proportions are when I want to mix together, and what temperature to heat it up to. And then I've played around a lot with different combinations of like amino acids. That's one of the ones I've kind of spent a lot more time kind of perfecting. So I got a couple little notebooks where I'll keep track of different mixtures I've come up with that seem to work well. Because once you get a good kind of ratio of two compounds that crystallize well together, then you can start like adding in different impurities or that kind of thing, and the impurities will kind of give other effects.
Yep. And we know the difference between science and screwing around is writing it down. So.
Yeah, I always feel bad when I don't take good notes. I'm like, oh, I'm a bad scientist.
Well, sometimes I can imagine you were like, oh, I forgot to write down what it was I did to get this image, and then you're kind of like, upset because you want to do it again, and you're like, ah, crap, I can't find that.
Or I didn't at least try to write some kind of mark on the slide to kind of let me know what the combination was if I forget what it is. Like if I go back to it a few months later, if it hasn't decayed or something. Yeah.
Let's take a look at another picture.
All right. See if I can do this without crashing.
Nope, it disappeared. There it goes.
Is that working?
Hold on. There we go. So this is kind of feathery, so this one's a little bit different definitely than the last one. The color scheme is different.
Sorry, did you say something? The audio kind of cut out for me. I had to reload the OBS
okay.
thing.
Yeah, I was just commenting how this one, it's definitely a different kind of image than the last one, and it must have gone through a different process.
Yeah, this one I made, I combined 3 different amino acids that I found on Amazon. I guess they sell them as like bodybuilding supplements. It's like beta alanine, glutamine, and taurine that you'd find in like your Red Bulls and stuff. So I mixed those up in a real specific proportion, and I kind of dissolved them in, we can call it maybe 30% alcohol, or it was some vodka. But and then I heated it up on the hot plate, and the alcohol helps the crystals to spread out nice and thin so they don't clump up, and it gives these kind of smooth patterns. And then I might have put a little bit of food coloring in it too, which kind of added some different colors to it, and the propylene glycol and the food coloring made it a little thicker while they were crystallizing, which kind of gave these cool wavy forms that I wouldn't normally get from this combination. I thought it looked kind of like seaweed or something.
Yeah, well, I think it's amazing that it's still crystals. I mean, we don't always think of crystals as looking like this. I mean, it looks like more biological, like it is feathers or seaweed or something. It's not, a chemical substance that's been crystallized out of solution, and like the differences in size and the shading and everything, it's kind of alien in a way, but at the same time familiar at the same, it's kind of like if Vinny is still here, it looks like something you'd find in Subnautica in that game, right? It's like the seaweed of another world. So and
I was pretty happy to get this kind of pattern. It's pretty unique. For usually crystals form more kind of crystally looking stuff, I guess you'd say.
Now how long do the crystals take to form? So like, and you're making this one, do you have like start the process and you have to wait like 5 minutes, 10 minutes, or is it?
It's pretty quick. I got the hot plate up to about 90 degrees Celsius, close to the boiling point of water. So once you get you kind of heat up the slide, kind of either put a drop on the slide of the crystal solution or smear it around in an interesting way. Sometimes that makes some cool things happen. And then, once the water goes, usually it starts crystallizing pretty quickly.
Right.
A lot of times, like, you'll get like dust or dirt and stuff on the slides, and that'll that'll start it forming a little faster there.
So is what they call that nucleation? Is that what that is?
Yeah.
I'm trying to think of the terminology.
Yeah,
now,
exactly.
Is it the same as like what I think crystals, because when I teach earth science, that the rate of cooling will affect the size of the crystal? So if you
yeah, definitely.
The same like thing at the microscopic level.
Yeah, if they start forming, you can always like turn the hot plate off or put it somewhere cooler, and that'll either, in this case, they'd speed up a lot, and then you turn the heat back up, and then the crystallization rate slows down. Some substances, it's kind of the opposite. But yeah, that can produce some interesting effects too if you keep alternating hot and cold, or yeah, all the same kind of things apply. I get a lot of like metallurgists and stuff commenting about like metal crystals, and they use terms I have no idea what they mean. I'm just like, oh, that's cool.
All I know is it makes good art.
Yeah, they're like, oh, this is a cool example of dendritic nucleation or something. I'm like, all right, what? Okay.
Right, because you're a biologist by trade, not a chemist or a metallurgist.
Yeah, I wasn't very good at chemistry in high school or college, so I was more of a chemistry enthusiast now, I guess. But
yep. Well, you're putting it to work now, and it goes to show you don't have to have a PhD in something to be able to successfully use it in some way.
No, I mean, there's so much information available online about all this stuff.
Yeah.
I do read research papers occasionally, just to, not that I understand all the results, but sometimes they'll give me interesting ideas of substances to use, or different techniques to try to get the crystals to start, or I don't know. The Wikipedia is always my friend too. It's got all the melting points and boiling points of substances listed and what they're used for and everything. That's kind of what I try to do on my Instagram, like put a little blurb underneath it to kind of give a little background, or try to tell like an interesting story of the history of the chemicals discovery or something like that too.
Oh yeah, add a little interest to it.
Yeah. Otherwise, it's just kind of abstract art, and some of them are cool to look at, but other ones, I don't know. I think it maybe helps to have a little background to it.
Yeah, and I think it depends on the person, how they appreciate, what it is. I mean, art is in that way. You could appreciate it just from a purely aesthetic value. You could appreciate it from more of a scientific angle as well. And I'm sure there are people that appreciate it from a photographic, realm, as far as microscopy, imagery and things like that. So, it's like anything, you can appreciate it in a multiples of ways. And I try to get at things from different angles to get that appreciation. It's kind of like everyone can read a book, but you might get different things out of that book.
Yeah, that's very true.
So we've seen two images so far that are different, for sure. All right, I'm excited to look at another one if we want to try to bring up a third. I mean, you've got probably troves of these, but I was on your Instagram the other day checking them all out, and there was endless numbers of really, that I could tell, of really good ones. So, and I think people here aren't necessarily going to just want to just listen to us talk about it, but also just kind of take a look as well. And if you have not been on Chris King's Instagram or followed him on Twitter yet, please do, to take a look at our artwork that he is doing.
Let's try this one. It didn't crash this time.
Let me get this. And so, like, this is another one that's different than the other two. It's almost kind of in between. We've got what I see. I got two-toned, and I like how it's kind of like threads that seem to be, you know, twirling around each other. You know, like pieces of yarn.
Almost right on the money there, actually. It's mouse muscle fibers under polarized light.
Yep. So the muscle is the red, and or like is the other end like the tendon, kind of like where it's attached.
Yeah, exactly. Or ligament. Get those mixed up. Yeah, it's like from a mouse leg, not to be graphic, but it's stained with that Congo Red stain I mentioned too, so it made the muscle cells show up more. So these are skeletal muscles, so you can actually see the striations and the black dots are the nuclei of the muscle cells on the periphery. Because it's skeletal muscle. I always thought this one looked really nice. But a lot of biological components also polarize nicely. Bone when you cut bone real thin, it'll glow nicely under polarized microscope. Muscle tissue, like the scar tissues and the amyloid plaques that people get from getting tissue damage build up over time. A lot of biological molecules have kind of a crystalline structure.
Right, because they have a repeating pattern of some kind, you know, is reminiscent of that crystalline structure, and you know, muscle fiber has that to it. It's a repeating, you know, protein. So I can, you know, definitely see that, and it's fine. I didn't even notice the nucleus in those until you mention it. And then I'm like, oh yeah. Then you can see it. It kind of pops out.
Kind of subtle.
Yeah. Now I was thinking of these. The lighting's got to be tough, as far as it. I mean, it can't just be straight down, and it can't be just straight up from the bottom, and it can't be from the side because the shadow would be too much. So is it just the polarized light? Do you ever shine from like the bottom, or, I mean, is that a real problem, or you struggle to get it right till you find it perfect, or are you pretty good at that now?
Yeah, I do. Usually it's kind of the straight on polarized light, which I can kind of adjust it a little bit, and then can kind of give an illusion of depth or shading. Crystals usually have, or substances that can be polarized, their crystal structure usually has what they call extinction axes, which there's some kind of geometric thing to it, but basically it'll either have like one dark line going through it or like a Maltese cross kind of pattern going on. And those kind of rotate around, so I can usually rotate the stage of the microscope to I can get the shading in the right spot. It kind of gives like impressions of light and dark, or sometimes like shadows on a mountain range, depending on the.
Right. So instead of moving the light, you move the sample itself. You move the slide.
Yeah.
Right. That would make sense. You kind of know where you want it, and then you move that to try to get the shade that you want from it.
Yeah. And the other thing I do sometimes is use it's not quite like dark field microscopy, but I've got a kind of semi-dark field or oblique lighting filters, I guess they call them. And that sort of, you could just put a quarter over top of your light source just so light gets around the edges, and then that'll bring light from the sides, which can both polarize the thing and kind of illuminate the edges of things, which kind of makes a neat effect. I've got one I can show you of that if you'd like.
Oh yeah. I just love looking at these. I mean, that is really like, yeesh. So talk about this one. It's kind of landscapey, but it's
Yeah, it's actually the same mixture as that kind of seaweedy looking one that we looked at earlier, but without the food coloring added into it. So yeah, it's three amino acids crystallized together, probably the same temperature. And then I use the oblique lighting to kind of illuminate the edges of each single crystal. The temperature maybe was different. It looks like it might have been. I don't know. I thought I kind of sort of had this sort of raindrop feathers. Somebody called it like unicorn tears on my Instagram, I think.
Yeah, and you can see every edge. I think, and I think that's what it's impressive. Like every little tiny edge that's there is illuminated.
Yeah. Like, and some of those are like cracks in the crystals too that form. Like, I guess like as they cool down, they form, and then when the temperature cools even more, they start to squeeze past the structure they made, and you get these kind of like stress fractures that sometimes they look kind of cool.
Now you mentioned the time. Is it sometimes? I mean, we mentioned how long it takes. Is it sometimes time dependent? Like if you're like looking at it and you miss that window of time where it looks perfect and then it changes.
Yeah, sometimes if it cools off too quickly, that can kind of ruin it depending on what you're going for. Like I like doing caffeine a lot, like melting that and then letting it recrystallize. But it cools down so quickly. If you just take it off the hot plate, it forms tons of cracks, which sometimes look cool, but sometimes you want kind of more smooth, flat planes of color. So sometimes I'll just turn the hot plate down a couple of degrees at a time, and then over like an hour or so, it kind of forms more like a more smooth picture. Yeah, it depends. Like some days I'm down in my workshop for like hours or something working on it. Other times I just try to do a couple quick ones after work. So
right. Now, do your pieces have actual titles? I mean, you joked that someone called this unicorn tears. I mean, but have you titled your art?
Doing titles. I haven't done any titles lately, but people usually, I got a few of my Instagram followers usually suggest titles for me. So if I make like Etsy listings or something, some of these will probably steal their titles. Thanks, guys. So. They always come up with good ones, I think. I don't know.
So you go on Etsy, you can make your coffee table book, where you can have like the picture and then like the ingredients and how it was made.
That's a good idea.
There you go. That was free. That's what I do.
Appreciate it. Thanks, Scott.
Free ideas. Let's take a look at another one.
Okay.
We got time. Like I said, I love looking at these. I like that everyone that's here viewing can take a look at these as well. And
let me do a different chemical this time. This is cool.
All right, this is one of the ones that to me looked a little iridescent. If that's what that actually is, iridescence that we're seeing. It's kind of like that oil sheen, right, that you get when you see oil on water and the sun's hitting it.
Yeah, exactly. So this is Malic Acid, which is like I've heard it called apple acid. It's like a sour flavoring in candy, and I think it's used in beer brewing too. So yeah, it's like a sour taste, kind of like a citric acid or something you find in like your sour candies. And this I just dissolved in, I think it was in alcohol, and then just let it evaporate and it crystallized. I tried melting it, but I didn't get these cool feathery patterns on it. So the iridescence is just like a lot of small changes in the thickness of the crystals.
Right. And I know in bubbles that thickness, and here's the science piece of this, right? It's at the nanoscale level. A lot of that change in that difference in thickness that causes
interference patterns.
Yeah, that look in it when you have like a soap bubble. So it's a very small change in thickness can cause that change in color.
Yeah, that's kind of how the polarized lighting technique works, actually, because you have like the two polarizers cross 90 degrees, and a polarizer takes light and makes it directional. So if you just have them both cross with nothing in between, it'll look black on the other end when you're looking through the eyepiece. But you put a crystal or something in there, and it sort of acts like a prism, and it bends some of the light passing through, and then it interferes with each other going through the top polarizer before it reaches your eyepiece, and you see these, I guess basically they're interference colors, kind of like you'd see with those iridescent soap bubbles you mentioned. They're an oil sheen. And then I think they used to use that as a way to kind of analyze mineral samples when they were like drilling for oil to see if there's oil-bearing minerals in the ground.
Makes sense.
Identify each one by, you just cut like a rock really thin and look at it through polarized light, and yeah, you get a result. They make really elaborate charts that show like all these different rainbow colors and where they match up with how thick the sample is, and then you get a refractive index that lets you identify compounds.
That's what the refractive index. That's kind of like one of the things I was trying to think of.
It's a fun word to throw around.
Make you sound really smart. No, diffraction pattern is good too.
Yeah, I mean,
that's a good word.
Those sound good words to me.
Whereas feathery like pattern. What was that?
Oh, what did you say?
I said feathery like pattern is not as fancy of a word choice.
No, that's kind of what we're seeing, I guess.
Yep.
You want me to throw up a video or something? Or?
Oh yeah, video would be awesome. This is going to be like your TikTok pretty soon. Is that what this is going to be?
I've been contemplating getting on there. Yeah. Let's see. Let's do.
All right, video time. Don't forget, if you're viewing and you're in chat, please put in questions or even comments, for either Chris or myself. All right, let me get a copy of this. Let's get this up here.
Yeah.
All right, so what are we seeing?
All right, so this is sodium benzoate, a type of liquid crystal. We're sort of watching it crystallize in the open air here in this first frame. I just kind of heated it up, and this is its liquid crystal state solidifying into a solid crystal. And these are similar to the compounds you'd have like in your cell phones and your tablets and stuff in the screen. You can control liquid crystals using heat, or electricity, magnetic fields, that sort of thing, to kind of rotate them. So like our cell phone screens basically have like a light source, like a layer of liquid crystals, and then a polarizer on top of it, and that combination kind of lets us get all these cool colors and things.
Yes, that's pretty.
I used to music and stuff cool. Too.
But... yeah. So those of you that are in the stream that didn't know, because I know some of my students are in there. That LCD is liquid crystal display. So we have a question here. Volcano Doc is wondering, what is the camera setup used on the scope?
Oh, should I just answer it? Like,
Yeah.
with this one, I just I had like a clamp for my cell phone to hook onto the microscope eyepiece. This is before I got like a fancier like DSLR camera. Now I have like I used to use my microscope at work, but then I ended up buying one to use at home, so I thought they might get upset paying me for do my hobby at work all the time. So now I have like a Nikon microscope, and then I have it's got like a trinocular head, so it's got like two eyepieces, and then there's like a camera port on top that's got a separate projection lens that focuses the image onto the sensor of the DSLR camera. This cell phone works fine too.
So I've done some cell phone stuff at school with our basic light microscope. Nothing with crystals.
You don't really need a fancy microscope to do any of this stuff. But, like, once I got into it, you kind of jump down that rabbit hole. You're like, well, I could get a nicer microscope and a fancy camera. So I just bought like slightly nicer microscope lenses for my microscope like a couple months ago. Like, he's got a flatter field and the edges are better in focus or something.
So what's interesting?
Make your pictures any better, but
you can at least psych yourself out there. What's fun is like when I have students and I tell them, because they're always surprised, like, I can use my cell phone in class. I'm like, seriously, if you want to take pictures of your science and then put it on your Instagram or your Snapchat, go ahead. Yeah. Especially if we're doing pond water and things like that,
Oh yeah, I love looking at
it's amazing how good you can just
pond water counts on Instagram and stuff. Those pictures are amazing. Diatoms and I put another video up, or are we out of time?
Yeah, well, we got as much time as you want, really. I'm on vacation right now, so I won't keep you here all day, though. But I think another video would be awesome.
I got an email from work that the lab's closed tomorrow because of a snowstorm, so I'm pretty excited.
Woo! All right. Stay home and do microscopy.
That's kind of my plan. Play some video games, but
yep. What games are you gonna be playing tomorrow?
I'm probably gonna try to do Breath of the Wild some more. I've been trying to get back into that. One of my buddies is really into it, and he's like, "Yeah, it's only like 80 hours long." I'm like, "I got like 10 hours in it now." It's fun though. I get some more shrines, get some more hearts, so I can get the Master Sword at least.
Cool. I'm not too familiar with it, but I'm writing it down. Volcano Doc is asking, any recommendations for an affordable polarized light scope with a Bertrand lens and a trinoc? And what's affordable?
Yeah, I mean, that's a very loaded question, but Amscope, it's like I don't know if it's a Chinese company or something, but they sell through Amazon, and it's not like going to be like the nicest possible microscope, but less than 10K. Oh, okay. Oh yeah, the Bertrand, and then well, if you're trying to spend a few thousand, I mean your Nikon. I don't know. The Olympus BH2 is a really popular one amongst some of my microscopy rivals on Instagram. But your Nikon Optifot and Labifot, you can probably get a fully loaded one of those for like $2,000 or $3,000 with the Bertrand lens and trinocular head that would be in good condition. Sorry, I got distracted. Talking about microscopes.
It's okay. You were answering a question for chat. That's what we do.
Yeah. I mean, if you can afford a Nikon Eclipse or something, they're pretty amazing. But you can get an Amscope, probably one of those fully loaded for like $1,000 or $1,500, and you get modern optics and everything. I kind of like the vintage stuff though; it looks kind of cool. So this is soy sauce from my Japanese takeout that I crystallized. I like all the little These are good examples of what I'm told are dendrites, like the long crystals that form like the little branches and stuff.
Yeah.
And this one's not polarized. It's I use what they call Reinberg Illumination, which is basically just putting like two colored circles over top of your light source on the bottom of the microscope, like one bigger one and then a smaller, maybe darker one inside it, and that kind of gives you different colors from the different angles and makes the image look a little more 3D. But salt's not polarizable. That's basically what you got in soy sauce, besides all the proteins and stuff from the fermented soybeans and algae.
Right, so these are just the salt crystals coming out of solution, and then the dendrite, and it's in the colors making it look
stuff
nice.
Makes it a little more regular salt would just be cubes.
Yeah.
I got another one.
All right. Just I should have put this out as like ASMR. Because it's kind of got that fulfilling quality to it. Like watching crystallization, it makes you feel good. At least for me. I don't know if anyone else in chat feels the same way here. About that.
Yeah, I usually put them to like music for like kind of a music video thing, and then there's a whole other thing is finding the right song to go with it. But
here we go.
So this video is elemental sulfur that I melted, and then I took a soldering iron and melted it again through the cover slip. So you can kind of see it as it lifts off, and then crystals form.
Wow.
I believe I rotate the stage at 1.2.
Now, is this pure sulfur, or are there like impurities that add a little bit of the texture to it, or you just don't know?
I don't know. It was sold to me as pure sulfur. It's just like little sulfur flakes in a baggie. Seem legit. It's probably not 100% pure though. It's nothing usually is unless you get like grade stuff.
This video is really cool. Like Volcano Doc is talking about it. It's like you could string a couple of these together with some synthwave on it or something, and
yeah,
you could just watch that.
Yeah, I had one video that I got a psychedelic rock band like paid me to put one of their songs to, and then they bought a bunch of my videos to use as backdrops on their sets out in LA or something.
There you go. That's cool.
I was pretty excited about that.
Because that's what this kind of like reminds me of, some of those late 90s alternative rock videos.
I think synthwave would be a perfect match for some of these. Thanks, Volcano Doc.
That is cool. All right, well, it is about time. We can leave this running. And I have some questions for you, as is a tradition on this show.
Yes, sir.
So I'm going to ask you 3 questions. And if you get 2 out of 3 right, I'll gift a sub to someone in chat tonight. And this is, of course, not about anything you know necessarily, since you do microscopy and photography of the very small. I thought I'd ask you about somebody that does landscape photography, and by the name of Ansel Adams. So we have 3 questions about Ansel Adams, and of course this is like where you're like, oh, I did a research paper on him as a child. And if you get 2 out of 3 right, we will give a gift sub out into the chat. So, oh, there you go. Yeah, that's a cool video too. All right. So, question number 1.
I'm ready.
You ready?
Yes.
All right. So, which of the following did Ansel Adams say about landscapes? All right, this is a quote. Was it A, landscapes are beautiful and eternal, except for seasons. Landscapes change with seasons. Or was it B, landscape photography is the supreme test of the photographer and often the supreme disappointment? Or was it C, landscapes, if not for them I'd be poor? Wait, I am poor. So is it A, they're beautifully eternal, B, they're a supreme disappointment, or C, I am poor?
I'm going to go with B, I think.
You are right. B is correct.
Sweet.
That is a correct quote. I thought the other ones were kind of funny. I made those up.
Okay.
And he was poor until the end
like
of his life.
He would have said.
He didn't
I know
become...
he lived in a shack by Yosemite National Park his whole life. So.
Yeah. So he actually, he didn't make a lot of money on his photographs until he was like in his 70s. So late bloomer for him. All right. Next question. You got one right so far. How did Ansel Adams break his nose? Was it A, during the San Francisco earthquake of 1906, when he fell into a garden wall during the earthquake? Was it B, on a photo shoot in Bozeman, Montana, where he got into a bar fight regarding his displeasure with the Nixon administration? Or was it C, during a photo shoot at Big Sur, California in 1935, where he tripped over a tree root and fell into a tree?
I'm going to go with C.
It was actually A, when he was a child living in San Francisco. He was in the earthquake and he fell into a wall and broke his nose. And the rest of his life he had problems with his nose. So interesting. When you think about the span of his life from like that 1906 all the way up into like the 80s, that's like it's quite the life. Question 3. What did Ansel do that he later regretted? Was it A, during the Reagan presidency, he told Reagan that he hated him and that Reagan had no knowledge of the environment? Was it B, that to help publicize a book of his photos, he had the negatives destroyed and then he limited the number of prints of the book that were made? Or was it C, he never finished formal schooling and only finished with an 8th grade education? So which of those 3 did he regret? Insulting Reagan, destroying the negatives of a book he made, or C, never finishing his formal schooling?
Not real sure. I think B.
It is B.
Oh, nice.
Good job. It is B. Yeah, so the C is also true and A is true as well, but he didn't regret those. He only regretted B.
Yeah, I didn't think he'd regret saying that to Reagan.
So he was a staunch environmentalist, and so he did not like Reagan. He didn't like Nixon either, but he did not like Reagan. So that is very very interesting. That's one of the things I enjoy making these questions every week. So
yeah, that was interesting.
So I'll be gifting a sub out to some lucky person in the chat. Way to go. There you go. Guess who got it? My wife. As she should. Good job. Use those emotes. Use those. So good job on those questions. Sometimes so the landscape photography, its own issues, but the microscope photography, it's got its own little pieces of fun. The best part about your job is you don't have to worry about being in the snow outside while you're doing it, or getting attacked by lions, or
you don't have to go nearly as far to find a nice landscape with landscape microscopy.
Yep. So very, very good. So this video is really cool too. I wasn't even watching it because I was doing the questions, but it's like shooting across the screen.
Yeah, it kind of mesmerizes me too.
So are you actually like moving? You're saying like you have that circular base, or you're like kind of shifting
you're
that
seeing
around
the actual stage move, so you can kind of see the colors change a little bit as it. I mean, I made really cool. I think I did like forward, and then I made it go in reverse in the second half of the video or something.
Yep, that is really.
Try to get artistic with it. I don't know. Yeah, you can kind of see how the colors shift when you rotate it. Is it
cool? Hey, Freckled Science, welcome into the chat. Well, what is a histologist? She wants to know.
Histologist is a person that studies tissue. I think histo is Latin for tissue or something like that. So basically, you either work in hospitals or research, and we take tissue samples from patients or research subjects and cut them into really thin slices. You kind of like, you can either do frozen tissue, or you just slice it and use it like a paintbrush to throw the tissue on a slide real quick and then look at it. You do like a 5-minute stain, look at it under the microscope and do a really quick diagnosis, or you do the more traditional processing it through different solvents and then impregnating the tissue with paraffin wax to harden it up. Then we make little, put it in little molds, make little square wax blocks, and then we cut those at about 4 or 5 microns thick, which usually gives you a nice single layer of cells. And then we stain the cells, usually with hematoxylin and eosin. The eosin stains the cytoplasm of the cell nice pink, and then the hematoxylins stains the nucleus of the cell purple. And you can basically, you look for big clumps of purple because that means there's lots of nuclei, which usually indicates white blood cells and bacteria and stuff. Oh, you follow my IG? Nice. She's talking to me.
Yep.
Talking to me.
Yep. I don't put anything on my Instagram. It exists, but I don't use it.
Yeah.
I was thinking of making an Instagram for science streams, not for my personal self. I have that, but I was going to make one for the channel. Do you have another one like the mouse muscle one, like a more histological image?
Yeah. Hold on.
We can take a look at that since there seems to be some curiosity.
Sure. All my fancy stains up there.
Here we go. There we go.
Yeah, so this is a colonic mucosa. It's like the lining of your colon or small. I mean, this is small intestine. I'm sorry. It's probably small intestine. But anyway, these so you can see the nuclei of the cells is usually the dark spots, and then the red's kind of like the cytoplasm. And then all the blues, mucus that the gut secretes to break down food and lubricate the passage of your food through your digestive tract. There's another section of it from the side. So this is like kind of like the transverse section. Like if you go like the length of it, took a slice through, this would be like what the wall of it looks like. You have kind of goblet cells going through here, and they all excrete this blue mucus. Then at the bottom, you've got a muscle muscle tissue that kind of contracts the colonic lining, and then the middle part's connective tissue. So this is a Movats Pentachrome stain. It takes like 2 days to do. It uses 5 different dyes to give you 5 different colors and the right kind of tissue. You can kind of see elastic around these portal veins. I don't know if I got a good one. Yeah. It's around the blood vessels. You can kind of see the elastic lining of the walls. No, I don't get to do confocal microscopy. I would love to, though. That stuff's amazing. My boss's, or my coworker's husband does it across the street at the National Institute of Health. Mass cytology. I'm not sure what mass cytology is. Sounds cool. Yeah, I'd like to use all the different kinds of microscopes eventually, but confocals are huge and cost like $100,000. So it's a little. One day.
You have to wait for your employer to get one. That's when you rent time at one instead of actually buying one.
Yeah, that's what they usually do. You just get like a couple hours at it to finish your research project. I think it like uses lasers to scan the whole sample, but it destroys it as it goes or something. Which is kind of cool.
That's pretty cool.
Yeah.
Yeah. There you go. Say Freckled Science is a chem person, so chemistry ones would be cool too. But whichever one you want to share with us, these are all pretty cool images.
This one's like a housefly eye. I stained with that same stain. We kind of caught it in the lab and did the processing on it. So you can sort of see the
was it one that escaped, or was it on the banana?
He wasn't fast enough. We were bored. So. But the blue part's kind of like the ommatidia, like the protein cone that focuses the light, and then it goes into like a more of a retina kind of structure. Yeah, I can do another.
So you had a question where they're asking about your main field of study. I know we went over that at the beginning of the stream, but
yeah, I went to college for just general biology, and then I just did temp work in various labs. I worked in a fruit fly genetics laboratory where I grew fruit flies and raised them for genetic studies. I fed fish, I cleaned mouse cages, did a lot of that for a while. And then I worked in a research pathology lab where we did research. We assisted the research and tried to keep diseases out of the research animal colonies. And then 2 years ago, I got a new job working at a hospital. So now I work with people instead of animals. So just kind of a switch. But also interesting, and I got to learn a lot of new chemical procedures and healthcare related terms I didn't know before. So. Sorry, it takes me so long. I put too many pictures in the folder for.
That's okay. That's why we're here. This one looks really cool.
Yeah, so this is another liquid crystal. This one's called, the short name's MBBA. It just stands for a bunch of chemistry names I don't remember. But basically, it's like a liquid crystal around room temperature, so it's used in LCD screens. And they usually mix a few different types of liquid crystals together to get the right kind of blend of properties for a good phone screen or something like that. First time I messed with liquid crystals, I broke a thermometer or a timer or something in the lab, and I pulled the screen apart and used alcohol to like extract the tiny bit of liquid crystal in there and messed with that. What are those stores? How are they? is it censoring a microscopy related word?
Well, she was talking about retarders. I had to allow it.
Gotcha.
As you said earlier, unfortunate word.
Yeah. Yeah, I use retarders in some of the images. My microscope's got a quarter wave and full wave retarder, so the quarter wave one kind of gives everything like a white background. It shifts the wavelengths of the light that pass through by like a quarter of a full wavelength, which is like 520 nanometers or something like that, I think. And then the other one's a full wave retarder, which shifts it about a full wavelength. And that kind of gives everything a magenta background. So I kind of use those, and sometimes I don't fully cross the polarizers because you can kind of get different color combinations if you rotate the things. And lately I've been taking like a polystyrene petri dish and sticking that in the light too, and that lets me kind of cancel out some of the colors and focus on things when I rotate it. I just try a lot of different stuff, and like, you know, you get different effects sometimes.
Mm-hmm. I mean, this effect, this is really cool.
Yeah, so this one's just straight liquid crystal on a slide. I think there's a cover slip on it, and the little dots, I think they're water droplets mixed into it. Or maybe, I know sometimes this is kind of getting beyond my realm of understanding, but there's these liquids called chiral liquids that have like a handed structure, kind of like a DNA molecule or something like that. And when you mix them into liquid crystals, the liquid crystal molecules kind of line up in certain arrangements with that. And that can make kind of interesting fingerprint-like patterns.
You've made like friends in the chat with Freckled Chemist, because she does chirality and things like this.
Yeah, I only have a vague understanding of it, so all right, cool.
She says you're right, so there you go.
Nice. Yeah, I follow a guy called Vance Williams that does like liquid crystal research up in Canada or something like that. His Instagram's pretty awesome too. So I talk to him sometimes, and he gives me tips or. Yeah. He's an awesome guy. Yeah, this one, the black lines are like what I told you about the extinction axes too. So with liquid crystals, they, instead of being like north-south, kind of east-west, like a compass, it can be all over the place. I don't know, they're really interesting. Okay, got it, defects. Yeah. Well, I think they're nice, even if they are defects. I got a video of the kind of thing too, if you want to pop that up.
Oh, sure.
Still got time.
More videos. All right.
Yeah, I like the videos. I think sometimes the videos help me explain what's going on because you can kind of see it happening.
And it's good for the viewers. They like to see.
Yeah. This is that same chemical sort of mixing into the chiral liquid I mentioned. It's just limonene. It's like a cleaning product. You got to get like just the D or the L one. So that's like the handedness left or D, I don't know.
Oh wow, that's cool.
This one is crossed. Yeah, it should all be just pure cross polarizers without the retarders in this video. My memory, anyway. I made it like a couple years ago, so apologies if I'm wrong. Usually, if I get the black background, it's just a cross polarizer. Yeah, I put it to like a techno soundtrack. Okay, so that when it turned white there, that's when I put the quarter wave retarder up.
All right, we
wish I had like a smoother.
We'll put some music in there.
But,
Hey, it's me, my favorite hoodie, and I need a
Try.
First, I woke up to a nice
Like 20 seconds.
Where's...
That is so like.
Yeah, so I think it's the motions, like me, like jamming on the cover slip with the thing to kind of get it to move back and forth.
You end up. Is it kind of like, you know, Bob Ross? It's like happy mistakes, right?
Yeah, pretty much. Freckled
all happy mistakes.
Yeah, it might not be chirality related. Yeah, there's the charts. What would determine the colors, the thickness of it. I mean, the thickness would be changing at the edge of this, because this is kind of like near the edge of the cover slip when I'm like squishing it, and it goes up and down. Makes kind of like a wave. Yeah, that's a good soundtrack for it. 70s sci-fi movie.
Yep. I'll put the shout out to the music.
Nice. Yeah, I've been listening to more synthwave stuff lately.
So in those defects, like the fingerprinty like fingers, those are from the chiral structures self-assembling.
Isn't it cool?
Wow.
Yeah, cholesteric crystals. The first liquid crystals were cholesterol molecules, actually. My research. So I kind of... Yeah. I think they were discovered by accident or something. The guy was like, oh, this looks weird. Like all the good things
mistakes.
in science.
All rough, happy mistakes. See, we could always all sit here and just watch this over and over again with the music. And
yeah, I do that.
Half an hour later, we would just be like, what happened? What's going on? All right, Frank will bring this back to Earth. They were called cholesteric.
Freckled Science. I'm gonna have you start writing captions for my Instagram post, I think. Fucking great.
Had 2 melting points.
Yeah. This makes me want to go mess with them some more.
Well, you got all day tomorrow now.
Yeah, I know. I have to be productive.
Get some stuff done. Cool things.
Overreacting. Alright, so this one, this is Stearic Acid, which isn't a liquid crystal, but it's like it's a waxy compound used in soap making. Mm-hmm. I made this video. I was trying to make something for our talk tonight, so I thought I'd share, but it's just like drops of the stuff crystallizing. Yeah. It melts at a pretty low temperature. Hippuric acid does a lot of those cool kind of like concentric circles too, and urea is always a good one. My cat actually, I got a new kitten a couple months ago, and he peed on some plastic, and I noticed that the pee had evaporated because I hadn't noticed it right away, and left some pretty interesting crystal patterns. So I need to stick those under the microscope. Actually, he's a good boy. He's just trying to help. Really? Onion has cholesteric structures in it. Are you saying the onion, the tissue of the onion has cholesteric liquid structures in it? Does that make sense?
Oh, the cellulose.
Very cool. That's cool. Oh, I could probably make that at home. So one of the ones we
Probably.
did in plant biology lab. Take the onion cells, look at them under the microscope.
And that's what we do. In my classes. Sometimes. Beetles form it. Leaves us some plants.
Very cool.
There you go. The biochemistry. More of it.
Yeah, if I could go back, I think I'd probably maybe try to get into a field like that or something. But it's fun just to enjoy it too. Physics. Oh. I don't know about physics.
I don't know about that. Now you're pushing it.
So there's a lot of math and physics. That was my problem with physics class. We can pretend it's chemistry, can't we?
I think it's kind of like you take what you need to know and you apply it. This is applied chemistry or
fair.
applied physics in a way, right?
Nice.
So, see, now that we got music going to this, it's like chill time.
Yeah.
Oh, here's another one. Look at this.
Yeah, here's one I did of, I wanted to do like a chemistry reaction under the microscope, so I used the light retarders to make the background kind of pinkish, but it doesn't really have any other polarized aspect to it. But it's just an iron wire I pulled out of something, I guess a picture frame hanging wire and some acetic acid and copper sulfate I mixed together. I don't know, I thought it was cool.
That's pretty cool.
Yeah. I use a Nikon Optifot 2 with the full polarizing kit. I got it from like an old geology professor that retired or something. It was in pretty good condition. It's got the trinocular head. I got a... Yeah, it was expensive.
Well, I was saying earlier, they're all kind of expensive. You know, it's just...
Yeah, I mean, I didn't know about the cheaper Chinese ones and stuff. And you could pretty much use any microscope for polarizing if you just stick some polarizing filters below and above your sample.
See, now I'm curious. I have a polarizer at school. I have microscopes. I could try to
just 3D print them. Yeah, I mean, you could just take a pair of polarized sunglasses and pop the lenses out, or buy some cheap polarizing film off Amazon.
No, I have like a tub of optics equipment, including polarizers.
Oh, nice.
So
yeah,
I've taught pretty much everything, so I have all kinds of equipment.
Yeah, transmission's not the best with the cheaper polarizers, it's true.
Yeah, they block a lot of it as well as polarize it.
Yeah, I mean, like when you're doing photography, you kind of want a higher light transmission because then you can use a lower ISO on your camera and then your picture looks better. Especially with video. Pictures, you can kind of just lower the shutter speed until you get something decent.
I do have some beetles. Yes, I have some beetles that are like in plastic resin. I have some beetles like that, so...
So this was
circularly polarized light.
I was fooling around with at work. I took some plants I found on my lunch break. I think this was like a pine needle or something like that, and I did a fluoroglucinol reaction to it. You just stain it with a compound called fluoroglucinol, and then put hydrochloric acid on it, and it reacts with the lignin, like the stiffer parts of the tissue, and turns them red. A little plant histology, I guess.
There we go. We're covering all the bases here for sciences with this stream tonight. We've hit botany, we have hit animal physiology and physics, a little bit of earth science.
Yeah.
We're everywhere.
So this one's like a dye. I read a paper that said some sorts of dyes, like I think this one's like tartrazine. They have like a liquid crystal state as they evaporate, so you kind of see like this wave of liquid crystal along the edge of the crystallization thing. Something to do with like the concentration of the dye.
Card can offer cash back, but only Discover matches all the cash back you've earned at the end of your.
Ooh, you know words I can use.
Can
I steal fluorescent from work? Not at this job. The last one I could have, yeah. Sorry if anybody from NIH is listening. We weren't using it anyway. It was like Kodak chemicals from the 60s. It was fine. Just kept everything. A bunch of hoarders.
Well, I don't think anyone from my district is listening that I still have a microscope from the old building when we moved, because they were going to demolish everything. So I was like, well, you're not going to demolish that. So it's like a microscope from the 60s I have in my room.
Oh, nice. Yeah, I do have like an old lights one from the 50s or 60s I picked up pretty cheap. It looks so nice. You got like the black enamel finished, and
this one has
made.
Yeah.
Yeah, I love the old ones. They all look so cool. Like I go on this. I kind of lurk in this Facebook group, like amateur microscopy. I just like looking at people's microscopes they pick up and stuff, and some of the things they photograph. But
well, they're built like it's like solid iron almost. You drop it, and put a hole in your floor, versus the plastic ones now, where you wouldn't dare.
Yeah, mine was kind of when they're starting to make more stuff out of plastic, so I got to be real careful with the fine focus thing, because those the gears tend to strip out or something.
Yep.
Otherwise, it's pretty solid. Yeah, I'm making like these too.
I will say, unfortunately, microscopy is not something that's easily done with students over the internet.
Yeah.
That is an unfortunate thing from the last year. There's a lot of experiments we can do, and microscope, it's a little tough. You do? I don't know how I can stick a microscope in my students' hands to actually use one. I'm not teaching that this year anyways, but so I haven't had to problem solve it.
Yeah. So this one I'm sticking a screwdriver through some plastic. We can sort of see it change color as it stretches. It's not the. I was just trying to show like the strain. I don't know, when you stretch plastic, atoms get strained or something like that, and then polarized light shows the colors changing. Not as visually stimulating as some of them.
Yeah. Oh yeah. Well, my thing is I can't use Zoom or OBS at school because my computers can't use it. We use Google Meet. That's it. So if I was at home, it'd be different. But so I'm stuck with what my district gives me, which is not OBS. So it's one of those things where my computers at home and stuff is nicer than the stuff at school. Oh, that one's cool.
Yeah, it's caffeine. It's crystallizing from solution. All those little needles stabbing your cells and making you all peppy.
Wow.
This one's Naproxen Sodium or Aleve, the painkiller crystallizing. I just crushed up a bunch of Aleve tablets and extracted it with alcohol. It's a good time.
Look up Bart Carr.
Bart Carr. All right. Will do.
It's all crystallization. I did see that recently. They had the smallest video taken of the beginning of salt crystallization. I forget what researchers did that, and you could basically see the initial salt crystal form from just like 8 atoms, I believe it was. It was like ridiculous.
That's crazy. I'd love to see that.
I'll have to put the music back on.
Yeah. Seems like a good one for the music.
There you go.
Sometimes dry. Yeah, sometimes if you let it go after they've seemingly dried up, it does some. Like I've tried doing like a mixture of like salicylic acid and malic acid, like salicylic acid from like acne medication, and like it'll crystallize one way as the solution evaporates and the crystals form. It'll look cool, and then like come back like in half an hour and like get all these malic acid crystals ruining everything, like growing over top of it. Yeah, sometimes they like, yeah, I guess the phase separate. I don't know the terms real well. Like, the salicylic acid just crystallizes way faster than the malic acid, but when they're both like in solution together, it seems like one interferes with the other one's crystals forming. Gotta mix tartaric acid. Like if I mix tartaric acid, L and D together, I've done it just tartaric acid L, I think. Took like 6 months to crystallize. I mean, to finish crystallizing. Oh, interesting. I'm learning a lot from this.
Say, she knows her stuff.
Which tartaric acid? Yeah, I probably did too much. I used to use like big ass cover slips. I was like, yeah, I'll put a lot on, but you don't really need like a tiny bit because you just want enough to fill the field of the microscope.
Mm-hmm.
Yeah, well, I was trying to get it under the cover slip. Yeah, some things crystallize a lot faster when they're exposed to air. Like polynol crystals form better when you like stick it in between 2 hot slides, melt it, and then pull them apart. You get all these cool like ridges and valleys and
Two years of academia.
That sounds kind of depressing. This is an amino acid combination. Glutamine and beta-alanine. Ooh, goddamn. That's rough.
That is. Yep, it is getting late. We're going to be finishing up here in a little bit. I know.
Yeah,
you have a snow day, but it is almost. We've been streaming for over an hour and a half. But like I said, because this stuff they could just like.
All right.
I'm sure everyone else that is here could just like look at it and just watch it. You know, watching these crystals form. There's something very therapeutic about it.
Pretty much gotten through all the videos I had queued up, so
that's really cool.
Thank you for having me.
Oh, thanks for being here. It was awesome. You know, I think this in particular, in some people ask like, oh, why don't you do like a podcast? I'm like, this wouldn't work on a podcast, right? This is the kind of
it wouldn't be nearly as exciting.
Right? It would, right? You have to kind of, you know, you got to see it, right? So I think you know, a stream is a perfect medium for, you know, getting the word out about, you know, what you do and the science behind it and the artistry that goes into it. Well, that was cool. Was that like a drop? I could almost like.
Yeah, that was cool. I was dissolving some of the ones I didn't like as much. So you can kind of see the alcohol evaporating out. It's like a 70% alcohol or something.
That's so cool. That's just really cool.
Thanks. That'd be a good one to end on since it's dissolving the crystals instead of making them.
That is.
Yeah, I don't know. I mean, podcasts are great, but it's nice to have a visual too.
Oh yeah. Yep. So definitely we will be in touch. I put out your information. Hopefully people will go and check out what it is, you know, that you do, especially on your Instagram, which is really good.
Want more followers.
You know, you could even, you know, get some prints if you so feel that you want. This is good stuff.
Nice wall decor.
Yep. And of course, now you have the idea. You're gonna get your coffee table book out with the
Thank you for that. Appreciate it.
Put that up on your Etsy there. So next week we're gonna have. I'll put this in the chat as well. Ansel will be here. She's another education streamer. She will be in here chatting. And it'll be another great chat, I hope. And please stop in again for that. Friday, we'll be playing. I plan on playing Subnautica again. So, and maybe Vinny will be here for that too. My student. He's not here anymore. He has better things to do than watch his teacher on a Wednesday night. But. It was great having everyone out. Now we'll do a raid. We got 10 people here. We'll see if anyone's around to raid right now. So we'll stop in to somebody here. Usually Rocket Sage is streaming, but usually she is finishing up. So I don't know. It looks like she's still chatting for right now though. So we'll see.
The nature of the world we live in, there's trade-offs. You know, there's
there's nothing perfect.
That is one of the advantages that you get with nuclear energy.
So we'll stop into there. So she's still going on there. And we'll probably be there for just a quick while, and then she'll stop over. So thank you everyone for hanging out here tonight, and thank you Chris for being here and all your awesome artwork. And we'll be seeing you around on Twitter. And take care.
Hey, thanks for letting me come on and share.
Hey, not a problem. Loved having you here, and I really can't get over how beautiful some of those were and how those turned out. That's just like amazing.
Thank you.
I think everyone else would agree. That's good stuff.
I hope so.
Hope so. But all right, let's get going with this raid here. And everyone stick around. Say hi to Rocket Sage, who is a supporter of this channel as well. We can support her where we can. And I'll see you later.
All right.
Continental.
So it's because the basin range is still spreading out.
Yep.
And then basically the idea is that they just pump water down and then heat it up.
I think so.
And then it comes back up to the surface hot, and then they can run the turbine off of it.
Yeah.
And so I know that there's new exploration here. So it makes sense in certain places, and it doesn't make sense in other places. You wouldn't put it in the middle of the Rockies.
Yes. No. There. No.
You know, because that's and so you just look for where does it work, and if it works, then you add it to the grid. And if all things are priced properly, the way that, you know, including all the costs of everything, then you'll know it's cheaper to make geothermal, and therefore it's probably better. So there was a question about, well, you don't have to cover the planet in reactors.
No.
So the amount of the footprint of a nuclear power plant